Okay, so we can start. Hello, everyone, and welcome to our third session of the Research Inside Transporter webinar series of 2022. My name is Jaymar Portillo. I am a marketing associate at Bazzone Technologies, and I will be your host for today's webinar. Today, we have two excellent speakers who will give us insight into the transport mechanisms of human antiporters. We are very happy to welcome Professor Edmund Kunji from Cambridge University and Professor Dirk Slotboom from Groningen University. So first, I have to give you some information. So please try to keep your camera off and the audio also off so that we avoid any interferences during the presentations. Also, be aware that this webinar is being recorded and will be available later on demand. And for your questions, you can use the chat option and write all your questions in there. We will collect them. And at the end of every presentation, I will read them to our presenters. A little overview how the webinar will go today. We will start with an introduction to the surfer technology with our application scientist, senior scientist, Andre Bazzone. We will be followed by the presentation of Professor Dr. Kunji. After that, the Q&A session, and then we continue with the presentation of Professor Dr. Slotboom. And we close with some words then. Okay, so without further ado, we can officially give start to our third session of the webinar series. So I hand over to my colleague, Andre Bazzone.
So now you should see my presentation, right? So let's start again. So I just want to provide some general introduction to the surfer technology. Surfer actually stands for Surface Electrogenic Event Reader. And first of all, the surfer is very different compared to conventional electrophysiology. As samples, instead of whole cells, we are using vesicles or protoliposome that are attached to the solid supported membrane, which itself is located on a gold-coated sensor, as shown here. So you actually have lots of different millions of liposomes, vesicles attached to the sensor, and these are measured at the same time. And this leads to the possibility to measure even slow transporters. So signal to noise is superior compared to patch clamp. We actually have capacitive sensors. So we are measuring charge movements across the sensor via capacitive coupling. How does the measurement work? We actually perform a fast solution exchange, providing the substrate. The charged substrate moves across the membrane, charging the sample membrane. And this in turn charges the measurement electrode below, which is measured. So our surfer devices are actually robotic pipetting instruments. So we basically do a fast solution exchange from a solution not containing the substrate. So we record a baseline current to solution containing the substrate. And this leads to the measurement of the transport activity. The measurement takes place at 0 millivolt. This is again in contrast to conventional electrophysiology, where usually voltage steps are used to trigger transport activity. During a typical experimental setting, you are measuring several conditions sequentially on the same sensor. As you can see here, a pH dependence of a proton-coupled sugar transporter. So repetitive measurements can be done. Different concentrations or compounds can be measured one by one. The high sensitivity of the sensor not only allows to measure transport currents, but you can actually also measure fast pre-steady state currents, such as conformational transitions or any kind of translocation, single translocation step. So the challenge here is to do the data interpretation and data analysis, since all the currents recorded on the surfer are transient, as shown here for a transport current and for a typical binding current. So you need to do careful control measurements to distinguish between transport and binding. This last slide just shows you the two different devices we have. First, there is the Surfer 96 SE, which is a higher throughput platform. This is using 96 sensor bell blades, so it can measure 96 sensors at the same time and is able to record about 10,000 data points per day. It also comes with a powerful data analysis software. On the other hand, we have the Surfer N1, which allows for more flexible assay design and is very easy to learn, so you can basically teach a student within a day. With that instrument, you can measure about 150 data points per day, and it is also commonly used as a device to actually develop an assay before transferring to the higher throughput instrument. And the studies by Dirk Slotboom and Edmund Kunji, which are presented right after my quick intro here, have actually used the Surfer N1. So with that, I would like to hand over back to Jema.
Thank you, Andre, for the nice overview over the Surfer technology. So now, without further ado, we will introduce our speaker, our first speaker, Professor Edmund Kunji. Professor Kunji became a tenure-track program leader at the MRC Mitochondrial Biology Unit in 2000s, and he was appointed Professor of Biophysics at the University of Cambridge in 2019. Previously, he studied physics and mathematics, chemistry and biology at the University of Groningen, and visual arts at the Art Academy Minerva in the Netherlands, where he obtained his master in biology and chemistry. He carried out his postgraduate studies at the University of Groningen, and he obtained a PhD in mathematics and natural sciences. Thereafter, he continued with his postdoc studies in the group of Richard Henderson at the Medical Research Council in Cambridge. In 2003, he received the ENBO Young Investigator Award, and in 2005, he was elected Fellow of the Trinity Hall. And last year, he was one of our Surfer Grant winners. So we are very happy to have you here today with us, and we hand over to you for the presentation.
Okay, great. Well, thank you very much for the kind introduction. Give us the opportunity to present our work here. I first would like, these are the members of my laboratory on the left-hand side, but in particular, I'm going to present the work by Bazzone. And I'm also gratefully acknowledge Roger Springer for interpretation of the data, which, by the way, isn't completed, as you will see. Of course, I also would like to be, I'm very grateful also, I'm sure I can advance this. Yeah, I'm also grateful to Nanion, of course, providing it with the grant that allowed us the use of the Surfer N1 that we use to collect our data. And they've been absolutely excellent in also assisting with technical issues and support in general. So I'm very grateful to Nanion. Okay, so I need a pretty extensive introduction to explain how we were interested in measuring charge movements across the membrane. And the protein that we work on extensively is called the ATP-ATP carrier. It's together with the phosphate carrier and ATP synthase are key components of oxidative phosphorylation. And what the ATP carrier does, it brings ADP into the mitochondrial matrix, and at the same time, the phosphate carrier brings in phosphate, and they migrate to a synthetic site in the ATP synthase, where ATP is then synthesized by rotation of the central stalk, and then by another rotation is removed from the catalytic site in ATP synthase. ATP is then bound by the ATP carrier and is transported out to the intermembrane space, and then via VDAC to the cytosol. Now we have to synthesize our own body weight in ATP every day. And that also means that both ADP and ATP need to be transported in such volumes. And it means each ATP molecule is actually synthesized, resynthesized 1,300 times a day, and it represents roughly a 1-minute cycle. So ATP molecule is used, and then it has to migrate back to the mitochondria and is resynthesized to ATP and comes back out for another cycle. And so it's a high level of transport activity that have to be continued every moment of the day. In the past, we solved 2 structures of the fungal ATP carrier in 2 different states. The first state is called the cytoplasmic open state. It's actually open to the intermembrane space in mitochondria. And the reason we could solve that structure is because it's inhibited by a very powerful inhibitor called carboxyaltotyloside. The other state is called the matrix open state. So now the central cavity is open to the mitochondrial matrix, and that state is fixed by a powerful inhibitor called valproic acid. And because we have these 2 extreme states of this transport, it allowed us essentially to work out how the carrier moves from one state to another. And to our surprise, it involved 6 structural elements. So this is one of the most complex transporters in terms of structural mechanism. Most transporters will have a 2-domain mechanism, but these are 6. And shown here, what we call the core domains are shown here in primary colors, and the gate elements are shown here in gray. And so these 6 elements move in a coordinated way to go from one state to another. And here this shows a little movie. It's a simple morph between the 2 states, which shows these 6 structure elements in actions. So on the left-hand side, you have the cytoplasmic view here. And you can see here that the opening and closing of the cytoplasmic side is achieved by the 3 gate elements shown here in gray. And then the opening and closing of the matrix side, which shows the matrix view here, is achieved by rotation of these core elements, C1, C2, and C3. And in this way, you have an alternating access mechanism where the central substrate binding site is alternatingly made available to one side of the membrane or the other. And this actually showed that the carrier functions as a monomer because there's no way that it can associate with anything else because of this incredibly complex mechanism. Okay, so now the thing that we didn't resolve really was how substrate binding allows the carrier to go from one state to another. And for this, we turned to a method that we used extensively called the CPM, and it stands for coumarin maleimide. And so the coumarin maleimide, the maleimide group reacts with the free thiol. And when it does so, the coumarin group becomes blue fluorescence. And so we can measure the amount of thiol residues that are become available. And so what we do is we purify the protein, and then we place it in a qPCR machine. And as we rotating the samples through a measuring window, we can monitor the blue fluorescence as we increase the temperature of the sample. And so at first, the cysteine residues are hidden, and then as the protein starts unfolding due to thermal denaturation, the thiols become available and react with the probe. And so what you obtain typically is this unfolding curve. It's a sigmoidal curve of which you can calculate the derivative. And then the inflection point or the peak point of the derivative we call the melting temperature. It's a representative temperature that gives a measure of the thermal stability of your population, where roughly 50% of your protein has unfolded. And it's great technique because if you change the conditions, for instance, you add something that makes the population more stable, you can get a shift in the thermal stability, and it means you get a shift towards the right. If you do something to destabilize it, it will shift to lower temperatures. So this is a means of measuring, for instance, interactions of molecules with your proteins. And this was the principle that we actually applied to our carrier. So if you purify the adipase carrier and add a range of different nucleotides, only the substrates ADP and ATP induce a shift in the thermal stability because they bind to the protein, introducing new interactions. And these new interactions make the population more stable. And you can do actually, as Vaso has done here, a concentration dependence of the thermal stability shift. And you see that if you increase the concentration, you'll see an ever increasing shift. And you can express it in another way here, where we show the shift, the delta Tm, here against the concentration of the substrate. And you see that you get an increase with increasing concentration till it hits a plateau. And at that point, you can't actually rescue the population anymore. The temperature now is too high to resist any unfolding, no matter how much substrate you have there. So it's not a kinetic curve at all. It's not a KD or something like that. It's essentially a reflection of the stability upon binding of the substrate. But then we actually argued if it's done through these extra interactions of the substrate with the protein, we could do the converse. And what we can do is maybe introduce mutations that abolish that interaction, and we should then see an effect on that shift. And so that was the basic premise. And so Vaso then created 36 different mutants of the translocation pathway. So these are all the residues that the substrate would meet going from one side of the membrane to the other. And she turned them all into alanine replacement mutants. So they're in total 36. And then when she measured the temperature shift in the presence of substrate at different concentrations, there were actually the vast majority of them showed the same shifts as before, so were not involved in substrate binding at all, except for 11 mutations. And so for the 11 mutations, we found that in the case of 5 of them, which are shown here in red at the top, you see that 5 of those mutations abolish this temperature-dependent shift, this concentration-dependent shift completely. And actually what it tells you is that these residues here are essential for substrate binding, where a single mutation completely abolishes binding. And you could see that, that all of them are positively charged residues in the central cavity. In addition, there are another 6 mutations shown here in orange, where the shift was still occurring in a concentration-dependent manner, but the size of the shift was highly reduced and significantly reduced. And so what it means is that these residues also have a role in substrate binding, but they're not essential for substrate binding. So it turns out of all the 36 mutations, only 11 of them are involved in substrate binding. And it turns out also, if you then do transport assays, measure it by using a radioactive exchange method, what Vaso could show is that for the 5 mutations that are deemed to be essential, indeed, the transport rate is completely abolished. And for the residues that are deemed to be important for binding, shown here in orange, you see that the some that are also essential for the transport function, but quite a few have maybe a reduced transport rate, but they're still active to some extent. So we think that they bind in like a cooperative in a manner that collectively that they are important for substrate binding. Right, and the other surprising result was when Bazzone measured this shift in the presence, and is shown at 10 millimolar, of ADP and ATP, she could show that these shifts were actually within error the same for ADP and ATP for all of these different mutations. And that actually tells you that ADP and ATP bind to the same site and with the same chemistry. So it turns out we could prove in this way that the adipate carrier has just a single site to which both substrate bind. Okay, and how could we explain it? Was when we looked at these essential and important residues in the context of the two structures we had, there was a very striking result. First of all, there are the residues which we dubbed the central substrate binding site, shown here. On this side, you get the residues that are important for binding, shown here in orange, which we believe bind the adenosine moiety. And opposite of it are three positively charged residues, which we think are interacting with the phosphate moiety. And when you look at those residues in the central binding site, they're pretty much in the same position in both states. So we think these are the residues that carry it for the substrate from one state to another. But in addition, there were four more residues, the role of which had not been explained previously. And we call them the asparagine arginine pairs. And there are two of them. One is above the substrate binding site, and the other one is below the binding site. And what was striking about those two pairs is that their conformations were actually dependent on the state of transport. So you have one here above the binding site that, first of all, in the cytoplasmic open state, point towards the opening of the cavity. And in the matrix open state, they become part of the substrate binding site. Conversely, in the matrix open state, the other arginine-asparagine pair point towards the opening of the cavity, while in the cytoplasmic state, they become part of the binding site. So in this kind of manner, we think that they're involved in guiding the substrate in and out of the binding site. And so they have a slightly different role. Now, so we can explain a little bit. Of course, this is not viewed in the context of the dynamic changes, but we have a fairly good idea now how substrates are bound in the adipate carrier. But when I always talk about the mechanism, and people that the hardcore bioenergenicists usually have a problem with our model that this is an adipate carrier that works as a monomer with a single substrate binding site. And this is the problem that they see. So across the mitochondrial inner membrane, you have a very high membrane potential, 180 to 200 millivolts. And if you place a carrier in that membrane that is exposed to this potential, the problem that they see is that in the import of ADP, which has three negative charges, you would have to move those negative charges against the membrane potential. And they would argue that this requires a tremendous amount of energy. And they then say that this must be coupled somehow to the export of ATP, because there what you do is move four minus charges towards the positive outside potential that they think works very well. So then they suggest to me that maybe you need some kind of spring loading of the protein where the ATP export kind of allows the unfavorable import of ADP. But it turns out this is not at all how this transporter works. It's not like that. They mentally think that this might be working as if it is some kind of channel, and that's not really what the carrier is. And so in order to understand a bit better what these charge movements really do, we turned actually to the machine that was already introduced by Andre, called the Server N1, which we very gratefully had access to via this grant. And Andre already explained how it works, but what it essentially does is you coat a gold electrode with a lipid membrane. And on that, you position a huge number of vesicles that contain your transport of interest. And what happens if the transporter transports, let's say, a negative charge? What happens is it creates a voltage change, and then the rate of voltage change is correlated to the capacitive current it induces via the capacitance. And so in this way, you can measure current movement or binding. And so when we did the experiment with the server, and these were done by Bazzone, and we repeated them a huge number of times, this is what you get. So you have your vesicles coated, and then what we do, they're empty on the inside, and we give them a pulse of the substrate on the outside. Let's say ADP and ATP. And what happens there is that, to maybe a surprise at first, if you give the transporter a -3 particle in the form of ADP, what you observe is a positive capacitance current. While if you give it ATP, what you find is a negative capacitance current. Now, you know that the formal charge difference between the two is 1. And so what you can do is you can integrate the total surface area underneath the two peaks, and it must represent one charge. And so then what you could do is divide them by the integrating of the surface area underneath. You can divide that one formal charge into two charges that have moved. And what you then get is a positive 0.4 charge movement when you have ADP being transported, and you get a negative 0.6 charge being moved when you have ATP moving. And so this at first might be striking, because obviously they're both negative particles, but the answer to this riddle is essentially the assumption that when ADP is transported, what happens is you have a 3.4 positive charge moving with the substrate when it's transported from one compartment to the other. And so if you get 3.3 positive charges -3 from the substrate, you get a positive 0.4. And if you now move ATP, you have a -4 particle plus 3.4 charges is a -0.6 charge movement. And so the way that the carrier really has solved this problem, it's if you want, it's given the electrophoretic advantage of ATP and passed it on to ADP, so that you have, even in the presence of the membrane potential, a stimulation of ADP import as well as a stimulation of ATP export. So this is how the carrier has evolved to deal with this problem of the membrane potential opposing translocation. It is by the central binding site also where the positive charges of the binding site move with the substrate. Now, another thing I want to point out here, and that these values that we've measured actually match beautifully well with the pioneering work of Brustovetsky in Ernst Bamberg's laboratory at the time, that actually this is the technique that brought eventually the Nonion devices. And they measured actually 0.35 and 0.33 at the time, which matches beautifully with the values that we are getting. And this was done for the bovine carrier. The one that we've done is for the fungal carrier. But it shows that the basic principles apply to both. Okay, the other point I want to make is that if you give it AMP, which is a compound we know is not transported, we observe something different, and that is that the peak is much shallower, and actually the duration of it is much longer. This takes almost about 200 milliseconds, while the transport peaks are only about 50 or even less than 50 milliseconds. And so, in essence, this is what we would say represents a binding peak that you see when you give it a compound that isn't transported. And this whole aspect of binding will come back later in a similar way. Right, okay, can we actually explain this 3.4 charge movement and read with the model that we had based on these thermostability shift assays actually quite nicely? Because you see here that the 3 positive charges of the central binding site move with the conformational change from one compartment to the other compartment. And so we think that this explains the 3 positive formal charges. At the same time, when you have a movement like that, you also move this arginine here, which is in this water-filled cavity in the cytoplasmic state, to the ceiling of the matrix open state. And we think that this represents this partial 0.4 charge. So we could think we can explain how these particular charges, charge movements. Okay, so, but can we actually prove that this is the case? Can we prove that these residues are involved in the charge movements? And so what we see here are the mutants that Bazzone generated of the 3 central positively charged residues. They were all turned into alanine. And now you see something very important. So first of all, to say that these capacitive currents that we see here are not due to transport, but they are due to binding, because as I've shown you before, these mutants are no longer capable of transport. And what you then typically get are these curves of longer duration, as we've also seen with AMP. But what's striking immediately is to see that the positive peak for ADP that you saw before has now turned negative, shown here in blue. And it means in essence that and the reason for it is, of course, that you've removed one positive charge from the binding site. So now you have a theoretical charge of plus 2.4. And if you now subtract 3 minus charges from that, you'll end up with a negative charge. And that's exactly what we see, in fact, for all 3 mutations. The other thing we see is that if you have ATP binding, you see that it induces a smaller but broader negative peak of it's not peak, it's a capacitance current, and which goes to 0 amplitude after about 200 milliseconds. For ADP, what we see is a sharper peak, a narrower peak, that actually is larger than that of ATP. And you see that it goes positive later. And actually, it reminded me of the binding profile that actually was shown by Andre earlier before. And then if you give it AMP, what you observe is that, for instance, here you see that this mutant still has a positive peak. And that makes sense again, because now you have 2.4 in the central binding site, minus 2 is still a positive peak. And that's what you observed. But you also observe that the total amplitude, if you compare the 3 mutants, is actually quite different. Now we know that there's a lot of variability between sensors and so forth. How much membranes have you absorbed to the surface? How many carriers have you successfully reconstituted? So we believe that there is a natural variation between different sensors, but we know that this is also quite reproducible. So in several different biological repeats, we see the same trend, and that the amplitude of the peaks actually decrease in a very specific manner, that you get the highest amplitudes for R287, a smaller one for K30, and an even smaller one for R88. And we think this has to do with their importance in binding of the substrate. And you see that very clearly for AMP, for instance, big peak here, smaller here, nearly all gone here. But it's also true for ADP and ATP binding. So these are generic effects on all substrates. Now, if you make those mutations if you change the mutations instead to alanine to glutamine, we get very, very similar results. It shows that these effects are kind of mutation independent. And again, also here you see that the ADP peak is now turned negative, again, for the reasons I've explained, which proves that these positive charge are involved in these charge compensations. Again, ATP peak produces a smaller but broader negative peak, which goes to 0 amplitude. The ADP peak induces a narrow peak, larger, but in this case does not go to positive as it was with the alanine mutations. The AMP peak, again, you can see here, gives a positive peak, so certainly for R278Q. But you can see that for the other two, the peak has gone now. And again, this is the same observations before, as that the amplitude is dependent on the mutation and differs in amplitude between different mutations. So we think that this actually means that the alpha-fold phosphate of AAP might bind to R88 or possibly to K30, but not to 287, because there you can see a large peak still. Okay, what about the two arginines in the arginine-asparagine pairs? And in fact, they show also current. These are much more difficult to handle. They're very unstable, so difficult to reconstitute. But Bazzone managed after a few times to actually obtain currents for those as well. And the first conclusion to be made is that though in both cases, the arginine to alanine mutation actually means that the ADP, which is -3, now induces a negative current, again showing and proving that these two arginines are also involved in the charge compensation by the binding site. And you see here that we still have to measure AMP to see what effect that gives. Now, if you change those two to glutamine, you see here again that they give negative peaks. And so these are a little bit more stable, easier to handle. But you see that in both cases, here now the ADP peak that was positive in the wild type is now gone negative, and it proves again that they are involved in substrate binding and charge neutralization. And you can see that both ADP binding and ATP binding actually induce a similar current. AMP2 is still to be measured. Okay, so how can we explain it? So this shows actually a close-up of all the residues we've identified to be important for substrate binding. And so what we have induced already in 2006, that you have what we call 3 contact points on helices 2, 4, and 6, which are the major way that the protein interacts with the substrate. And you see that 2 out of 3 positive charge residues in the central binding site are these contact points, and so indicating their importance, with K30 in between. So this is the arrangement in the central binding site. And then, by the way, I forgot to say, let's go back. Opposite of that, here, this here is where we think the adenosine binds, opposite of those 3 positive charge residues. Then above the binding site, you've got the arginine-asparagine pair, and below the binding site, difficult to appreciate in this view, you have the other arginine-asparagine pair. So these are the residues that are involved in substrate binding. And so we think that the way that the substrate now binds, based on those measurements, is that the adenosine binds here, as we said, and we think that the R88 is involved in binding of the alpha phosphate, and then the other phosphate groups of ATP will then bind to the other residues here, including a partial charge movement with the pair that is below here. So this is roughly how we believe the arrangement is. And this brings me to the end of my presentation. So we've seen about ADP carriers, that they're really pretty amazing. They keep us alive every moment of our lives, for all of our lives. They're among the smallest transporters in nature, only about 30 kilodaltons, and they only have 6 transmembrane alpha helices, but they transport some of the largest molecules, like ADP and ATP, and they do so without significant proton leak. They've evolved to have a completely unique transport mechanism, not replicated anywhere else, that uses 6 rotating elements around the central translocation pathway, making them the most dynamic of all secondary transport proteins to date. They have a single substrate binding site at the center, and 2 arginine-asparagine pairs that are involved in binding of both ADP and ATP. We have learned from using the Nanion server that there are 3 positive charges, 3.4 positive charges in this site, that are involved in charge neutralization that then stimulate both ADP import and ATP export in a 180 millivolt potential. And we've also proven by doing the mutagenesis that all 5 essential and positive charge residues are involved in charge compensation. And we think that 3 of those come from the central binding site, and that the 0.4 charge comes from the arginine-asparagine pair that is involved in the movement in one direction. And then the alpha phosphate moiety, we believe, could be bound to R88 because that's the most devastating mutation for the binding of all nucleotides, possibly also K30, but certainly not R287, which seems to always give the largest peaks upon binding. So I thank you very much for your attention, and I'm happy to answer any questions. Thank you.
Thanks for the nice overview of your results, Edmund. We have some questions coming through the chat, but because we're a little working with the time, so we will read then one of them, and the rest we will collect in a transcript. We will send it to the speakers, and then later they will be sent together with the on-demand link to all of you.
Yeah, sorry about that.
No problem. It was really interesting, so no way to miss that. Okay, so the first question that came here. Okay, so why are the peaks, the peak currents of the mutants for the ATP less negative than the peak for the ADP currents? Is it this result different from apparent affinity?
Right, okay, so I think if you do the total, the dynamics of them are very different, right? So you have a sharp peak for ADP and you have a very broad peak for, and so we think that if you do by and large, the integration shows they're about the same. So upon binding, the pattern seems to be so that this difference in formal charge does not matter. And it kind of suggests that in the way they bind, one of the charges is actually ignored. And that makes sense also from the thermostability shift assays, because ADP and ATP bind, if you want, induce the similar kind of stabilization. And so it kind of, both the thermostability shift assays and these measurements seem to hold up, show this particular effect, that the chemistry is the same. Now, in translocation, it's different because now you have to actually physically translocate a whole negative charge more in the form of ATP. So binding and translocation are not the same thing. So this is how we explain it.
Okay. Yeah, thank you. So there were some other questions coming up, but we will send them those to you.
Right. Thank you.
Okay, so before we move forward with our next speaker, I want to remind you that we have, so this is the third session of the webinar series. So I will put a link shortly in the chat where you can access the playlist and you can find the previous sessions for 2022 and 2021. Okay, so our next speaker is Professor Dirk Slotboom. Since 2021, he's a scientific director of the Groningen Biomolecular Sciences and Biotechnology Institute at the University of Groningen, where he also appointed a professor in biochemistry in 2014. He has a master in chemistry and biochemistry, and in 2001, he received his PhD from the University of Groningen as well. Thereafter, Dirk also continued with his postdoc studies, and nice coincidence, he was actually working together with Professor Edmund Kunji at some point. So we're really excited to have you also here. So without further ado, you can share your screen and tell us about your interesting results.
Thank you very much for this kind introduction. Yes, we were just discussing before this meeting that it was 20 years ago that I was in Edmund's lab, which was a great time, and it's great to see how the work on the mitochondrial carriers has evolved since then. So Edmund said that he was presenting some data which were not complete. The data that I will present are even less complete. So we focused on some of the work we've done with the server that we bought last year. So this is certainly not a complete story, and it's also I will not talk much about the human antiporters, which I had hoped I would talk about because we are not that far with those transporters yet. But still, it will give you a flavor of the work we do with the server machine. Now, the family of proteins that I will present about today are the glutamate transporters, and in mammalians, in the mammalian central nervous system, these glutamate transporters, they take up the excitatory neurotransmitter glutamate. So they are present in basically all the cells around the synapse, and if a nerve impulse hits the end of the presynaptic cell, then the glutamate is released, activates receptors on the postsynaptic cell, and the signal may be propagated. The glutamate that's released is then removed from the extracellular fluid by transporters in those cells, and these are ion-coupled transporters. And the mammalian ones, they transport the glutamate in symport with 3 sodium ions with 1 proton, and in counterport with 1 potassium ion. And by harnessing the free energy stored in all these gradients, they can reach glutamate gradients across the membrane of about 10 to the 6th. Now, this is work that was initiated a long time ago, and a lot is known about the ion coupling in these transporters by seminal work from many people. Now, in humans, you find 5 different subtypes of these transporters, and they all have slightly different functionality. They are also in different cell types and in different tissues, but they all can do the transport of glutamate with the 3 sodium ions, the 1 proton, and the counter transport of a potassium ion, and they can do accumulation of the glutamate. In humans, you also find 2 family members which are neutral amino acid transporters, and they are obligate exchangers. So they exchange 1 amino acid for another one, and that leads to sodium-dependent harmonization of the amino acid pools. I'd love to talk about these transporters today and their, well, let's say their electrogenic properties, but we are not that far yet. Now, both of these subtypes also have something special, and that was discovered by a couple of papers in the 1990s already, that apart from this coupled transport of glutamate with cations, there is also an uncoupled anion channel activity. And so these transporters, they can, well, they can act as, let's say, glutamate-gated or sodium-gated channels for chloride, which is probably the physiologically relevant one, but also other anions like nitrate and thiocyanate. Now, probably these channel activities are linked to chloride homeostasis, although it's still not completely clear what the function is. Now, you find these channel activities in the transporters I mentioned in the central nervous system, but also these neutral amino acid transporters, they have these anion channel activity. Now, I will today move away from the mammalian transporters, and I will present work on prokaryotic homologs of these transporters. And they have been, well, they've been discovered, well, even before the mammalian ones in some cases. Oh, that's not quite right. That's they were discovered separately, and then at some stage it was realized that they belong to the same family. And well, the 2 upper ones I show here, they've been characterized very well. The GLTPH, that was one for which the first structures by Erik Guo and Olga Butker were published. And this one is similar to the upper one, but, well, in our hands is biochemically a bit easier to handle. Now, these transporters have very similar properties to the mammalian ones, but in terms of cation coupling, they are simpler. So the archaeal ones, Pyrococcus and Thermococcus, they co-transport aspartate, not glutamate, with 3 sodium ions, and they don't co-transported with a proton or counter-transported with a potassium ion. And some of the other ones, they are not sodium coupled, but they are proton coupled, but I won't talk about that. Now, back in 2007, work from Joe Mendel and Renee Ryan already showed that GLTPH, the upper one here, most likely also has this uncoupled anion channel activity. Now, the experiments they did, they were fantastic, but they are very hard to reproduce in other transporters. So we've tried with a couple of them, and it was very difficult to do this. It's differences when you add valinomycin or don't add valinomycin, and you look at the level of accumulation. So we've been a bit unsure whether this uncoupled chloride conductance in the prokaryotic homologs is something which is conserved or not. And if yes, if it's relevant for anything or not. Now, so those are the transporters I will talk about today. I will now first give a bit of an overview of how we believe that this chloride conductance is connected to the transport activity of these proteins, and then I'll show the data we obtained on the server machine. Now, at the moment, I think there are well over 30 structures in the databases of these transporters, and it is actually now reasonably well known. Well, let's say in global terms how they work. So they are all trimeric proteins. They have two domains: a transport domain, which is on the periphery, and a scaffold domain, which forms all the contacts within the trimer. On the right-hand side, you see a top view from the outside of the membrane, and on the left-hand side, you see a side view. And these proteins are, well, they operate via a so-called elevating mechanism in which the three protomers in the trimer operate independently, and the transport domain can move up and down through the membrane as an elevator. And when it's in an outward-facing state, it can open a gate and then bind the three sodium ions and the aspartate. Gate can close, elevator can go down. On the inside, same gate can open, the cargo is released. Gate can close again, and the empty carrier can move upward. Now, this is reasonably well known, not only from structures, but also from all sorts of biophysical and biochemical assays. Now, the way that we now think that the chloride conductance is related to this transport activity is that at the interface between the scaffold and the transport domain, during the movement of this transport domain, transiently channel opens, which can conduct chloride ions or nitrate or thiocyanate, these negatively charged ions. And there's work from both molecular dynamic simulations from Jean-Philippe Martens, and also from the lab of Renee Ryan from cross-linked mutants structural information that indeed such a channel opens during the movement of this transport domain. Now, what also is relevant for what I will present on the data from the server is the kinetic mechanism of transport, the order in which the ions bind to this transporter. And the order is well established now. So it starts with a very slow binding step in which sodium is bound, and there's a conformational selection involved here, and that makes it very slow. First, two sodium ions bind. The first one is probably the slowest, and then the second one binds, and you get a transporter associated with two sodium ions, and that brings it in a conformation which has a very high affinity for aspartate, and that leads to fast aspartate binding. And upon the binding of aspartate, then there is a third sodium binding site, which now is of high affinity, which is then bound immediately, and then the transport step can take place. Now, this order of binding is relevant, and I will show you in a few slides why also the server might be able to to help understanding this kinetic mechanism of transport. Now, if you look at in a scheme in which we now present the states which we think are present during the transport cycle. On the left-hand side, you see the apoprotein, in which there's none of the substrates is bound. and it can move between an inward-facing or inward-oriented state with the elevator in the down position. It can go to an intermediate position and to the outward-facing position. There, the gate can open. This red line that's there. The two sodium ions can bind, but with the two sodium ions alone, the gate cannot close, so the elevator cannot move anymore. Only upon binding of the aspartate and the last sodium ion, the gate can close, and it can move again as an elevator to the inward-facing state, where the same gate can open again. And because of the low concentrations of sodium and aspartate on the inside, or low concentration of sodium on the inside, at least the goodies will be released, and the apo transporter can move up again. So it's the gradients it's the gradient of sodium ion that drives this in a particular direction, and also, of course, the membrane potential could play a role here, although we don't know which step actually is sensitive to the membrane voltage. Now, the questions that we asked really when we started working with the server, actually to play around with the machine, is whether the uncoupled anion conductance is conserved in prokaryotic transporters. Then if yes, and the spoiler is yes, it is, what is the physiological role of such conductance? And that question we still cannot answer, whereas in mammalian cells, it's probably related to chloride homeostasis. I find it unlikely that it would have the same role in the prokaryotic transporters. So maybe it's just inherent to the transport mechanism. And then we also asked, what is the structural basis for altered ion conductance in mutant proteins that are known, where the mutations are known from human diseases? So I will address all these points and then in the context of what we did with the server machine. Now, let me just start with presenting work on whether we can measure the coupled transport of the 3 sodium ions with the aspartate by GLTTK, the protein that is related to GLTPH, but it is a different homolog. Now, of course, this will lead to the transport of 2 positive charges, and the way we run these experiments is that we do it in a double solution exchange. We go from a resting solution in which there's potassium ions on either side of the membrane, so the protein is supposed to be in the apo state. Then in the first solution exchange, we add only the sodium ions, and then you usually get a solution exchange artifact because of the reconnection of the valves. And then in the second exchange, this is when we go to the activating solution, we add the aspartate, and then we see the capacitive current from the transport, the coupled transport of sodium and aspartate. And then we move to sodium again, and then see this oppositely, the current with the opposite sign, which is much smaller here, which is related to probably to the amount of transport that had taken place in the first step. And then we go back to the resting solution. Now we had to optimize this because regardless of how sensitive the machine is, we need a lot of protein to actually see these currents. And one of the things we did, we optimized the lipid to protein ratio. And then what you can see here is that this time decay of the capacitive current actually changes with the protein to lipid ratio that we use. So when we use a higher lipid to protein, so more lipid, less protein in there, then you see that the time constant gets slower. And you see that on the left-hand side, you see it quantified over here. And usually this shows that you are dealing with transport rather than just a binding event. So that was actually reassuring to us. We also played around with the absolute amounts of liposomes that we added there, and you can see that when we add more, we see more signal. Now in this experiment, we then varied the concentration of sodium in the experiment in the activating solution and also in the non-activating solution. And what you can see in the middle here, if you increase the amount of sodium, the current amplitudes go up, and you can plot this in relation between the current amplitude and the sodium concentration. And from that, you can determine an apparent Km of transport, an apparent Km for sodium at a fixed concentration of aspartate, which was, well, around 12 millimolar, which is great because this is very similar to the values we found from radio-labeled transport experiments. So it shows that the SSM machine, well, gives, in that respect, very similar kinetic data. There's also something else that you can see here. If you look at the decay of the current, then if you go up to higher sodium concentrations, the peaks get sharper. And well, there are multiple ways that you can interpret this data, but one of them is that because of the very low affinity for sodium binding to the apoprotein, when you are at low sodium ion concentrations, you have not saturated the transporter yet with sodium, and then the transport gets much slower because, of course, you have primed some of the binding sites for aspartate binding, but many of the binding sites then first need to go through this well, conformational change upon sodium binding. Whereas with the very high sodium concentrations, you don't have this, and you get this different kinetics. We interpret it this way now, but we are not completely sure about it yet. So as I said, we find an apparent Km for sodium at fixed aspartate concentration of 100 micromolar of 12 millimolar, and this is very similar to what we find in very classical radio-labeled uptake experiment where we have proteoliposomes and radio-labeled aspartate, and we simply measure the rates of uptake at different sodium concentrations. And the purple one over here is the one at 100 micromolar aspartate, and that looks virtually identical to the SSM curve. Now then, we showed that in the absence of any of these permeant anions, we can actually measure the sodium coupled transport of aspartate. But now we also wondered, can we measure the anion current? And for that, we used a different protocol of measuring, again, a double exchange protocol. But in this case, we start with, in the first solution exchange where you see the artifact, we already go to sodium plus aspartate. And then in the activating solution, there is one of these permeant anions. And we chose thiocyanate because that is the most permeant one, which is very similar to what you find in the mammalian transporters. More permeant than nitrate, and with chloride we see actually hardly any signal at the moment. So now you see, instead of the positive current, you see a negative current, because what you're measuring here is the anionic current from the anion channel. Now there we did a very similar experiment in which we changed the sodium concentration again between 0 and 300 millimolar, and from that we, of course, could also determine an apparent Km for sodium ions. And again, this is very, very similar to what we found in the previous measurements and also in the radio-labeled transport measurements. So this gives us confidence that the signals we see here are indeed for the anion conductance related to the transport by this protein. Now then, let's now that we can measure this, let's go to a mutation. So there are several mutations that have been found in mammalian glutamate transporters that affect the chloride conductance. And one of them is a mutation in EAT1, proline to arginine mutation, which leads to episodic ataxia, which is an hereditary disease. Now, what you can see in these graphs, this is a paper from 2012, is that the anion conductance in this mutant protein is much larger than in the wild-type protein. So in the mutant one, the open symbols, it's much larger than in the wild-type protein, the closed symbols. And also, which is you cannot see here, but also the transport activity of glutamate is lower. So higher anion conductance, lower coupled transport activity. Now this mutant is located in in one of the transmembrane helices of the scaffold domain. So it's in transmembrane segment 5 in the structure. I've cut out one of the 3 subunits here for clarity, but it's over here. It is really at the interface between the scaffold domain and the transport domain. And this proline is conserved throughout the family. So not only in the mammalian transporters, but also in the prokaryotic ones, a proline is located in exactly the same position. And in GLTTK, the protein that I will talk about, it is the proline at position 208. Now, we wondered whether we could, well, on the one hand, reproduce the effect of this mutation that was found in the mammalian transporter in the prokaryotic transporter. But we also thought that if we can do so, then it's very straightforward to determine structures of this protein, that we might also be able to see what causes this lower uptake of glutamate and higher anion conductance. And there were many theories on how this would work. One of them that this mutation would destroy the binding site, yet still allow the chloride conductance. So that's the way we proceeded. And so we made this mutation, and we measured with the SSM machine both the aspartate induced currents from only the sodium aspartate symport, so in the absence of the anions, and also the thiocyanate conductance, so in the presence of sodium aspartate and the permeant anion. And again, we could measure these sodium dependency of the amplitudes that we measure here, and we found indeed that the transport of aspartate, the coupled transport of aspartate was reduced, but the anion conductance was not affected. Now, I should say here that it's very difficult to compare, let's say, batches of liposomes, as Edmund also indicated. There's when you add them to the machine on the chip, it's difficult to quantify exactly how much protein you have there. And it also links to reconstitution efficiencies in the proteoliposomes. So we're now trying to quantify better how much protein we have on the in the machine. But at least what we can do is now to compare the relative amplitudes, and clearly the anion conductance relative to the aspartate uptake activity is about 4.5 fold higher in the mutant, which is similar to what was found for the mammalian transporters, at least qualitatively. Now, having found that indeed this mutant had a similar effect, we then moved on to single particle cryo-EM, and we solved a structure in lipid nanodisk of this mutant. And well, disappointingly, it didn't look any different from the wild type protein or other mutants that we had determined structures of in the past. So nothing major happened in the region of this proline to arginine mutation. The protein looked exactly the same still, apart from one difference. So in this really very difficult to read figure on the left-hand side, there are these red densities that you see here, and these are lipid molecules that we can find in our cryo-EM densities. And there's one phospholipid that actually forms a salt bridge with the arginine that is introduced at the position of the proline. And what we think is that by binding this phospholipid, it may affect the movement of the transport domain for which this lipid would have to be dislocated, and it may allow the protein to linger for longer in a particular state that indeed is conductive to anions, but because it lingers longer in this particular state, it will affect the coupled transport of sodium and aspartate. So this is the hypothesis we have now. We still need to work on this. So to conclude, in our hands, the SSM could faithfully reproduce the kinetic parameters of aspartate transport that we had obtained from classical radio-labeled substrate transport. We could confirm that also GLTTK, the prokaryotic transporter GLTTK, has an anion channel activity. The mutation that in E81 leads to diseases, also in GLTTK leads to increased anion conductance relative to the aspartate transport. And well, we still don't know whether there's also an absolute increase, but at least relatively it increases. And from the structure that we determined, well, we have at least an hypothesis of how the mutation might affect the transport and ion conductance activity. Now, with that, I come to the end, and I'm only going to acknowledge 4 people who were involved in this project. It is a long-standing collaboration with Professor Guskov from our university. Emanuela Colucci is a PhD student who did most of the SSM work that I presented today. Mijer Patino Ruiz has been an absolute, Mijer comes from the lab of Klaus Fendler, and so without him, I'm sure we would not have managed to get here. So he's a postdoc in Bert Polman's group, and I'm very happy that he's around here. And also Zaid helped enormously together with Emanuela to collect the data on this. And that's all. So thanks for your attention.
Thank you, Dirk, for the interesting overview of your results. We have some questions here for you. So the first one is regarding the last part that you showed. So this lipid bridge between, the bridge between the arginine and the lipids. Would you have more information about that? So have you done, for example, experiments with different lipid compositions?
Well, this is an excellent question. This is, of course, on our list of things to do. So far, we have reconstituted it in E. coli polar lipid extract with a bit of PC. So we need to go to specific lipid compositions now. Yes, okay. But we haven't done it yet. Okay, perfect.
Okay. Do you control the protein orientation in the liposomes, and how this can influence the currents that you see?
Yeah, this is, well, actually, this question I also asked to Edmund, but there was no time to do. So in the experiments that I showed, we don't control the orientation, and so they are probably mixed orientation. We have ways to do so. So we have raised synthetic nanobodies that specifically inactivate one of the 2 orientations, and so we can do this. We haven't done it yet. These proteins, they are, well, bidirectional. So of course it can affect the kinetics to some extent, but I think overall it will not make such a difference in whether there is chloride conductance or not. So I think in both orientations we would be able to see chloride conductance upon transport. But it's a good point, and it's something that we certainly will figure out.
Okay. Thank you. So there are some other questions also coming through the chat, but also for time reasons, then we will stop it here. So we will send you the questions, and then you can answer to them, and we will send it to the participants from today. So yeah, I would like to thank you both, Edmund and Dirk, for having this great talks today, which was actually a wonderful way to finish 2022, the webinar sessions for this year. Thank you very much for participating here with us. For the participants, you will get then in the upcoming days an on-demand link to access this webinar together with a transcript of the Q&A session. And if you if another question comes to your mind and so that was not written here in the chat, you can always write to us, and we will direct them to the presenters. So thank you very much for being here. and we look forward to see you for 2023.